as a layperson virologist, it's important to understand that RNA, when it's found in the wild, is never found in a high quantity. It's not found in a high purity. It's found in an incredible array of noise, almost inestimable, because you're talking about a single molecule that you're looking for, and whether or not there are 10 copies or 100 copies, there are not millions of copies, and none of them, by virology's own admission, are identical. In fact, somehow or another, they wax intellectual about how identical genomes would be a problem for viruses. They need to mutate all the time. But these contradictory ideas end up just kind of, they just kind of destroy themselves when you take them to their logical conclusion. But what happens in any RNA paper is, this is the trick. They find this sequence, and I apologize, my pen is probably out of ink. They find this sequence in the wild, and what they do is that they reconstruct it. They just make it. So if they, for example, you look at any of these papers that, and this is where I learned it, how I learned it is going back to the papers with Barrick and Shengli Shi, and you look what they did. They will find sequences using any technique you want. I don't care. You use primers aimed at conserved regions, or you use metagenomic sequencing that is preset to find coronavirus sequences that you have already predetermined are what you're looking for. And they find those sequences and they synthetically make them. And I draw a circle like this with a little arrow here because that's what these molecular biologists draw as a little plasmid. They make a DNA plasmid of it, and then using any number of commercial means, they make a lot of that DNA that represents the original RNA that they claim to have found in the wild. And now, depending on how they want to do the actual virology experiment, they could take this DNA and make it RNA first, and then put that on a cell culture, and that would be transfecting a cell culture. Or they could take this DNA and they could package it in an adenovirus or another kind of virus, and they could transform, because there is slightly different language for using a viral vector and DNA to change a cell culture. This is transformation. And using RNA to do the same thing, and that's transfection. So technically speaking, if you wanted to look at the COVID interventions, the adenovirus should have been called a transformation, and the RNA would have been called a transfection. But already going back to Jesse Gelsinger, they haven't used the correct words. When Jesse Gelsinger died, they called them gene therapies, just like they did now in 2020. So my point would be is that virology itself is based on this false equivalence. Now, why is this a false equivalence? Because if I use commercial or laboratory recombinant DNA to make this RNA or to transform these cells with DNA, I am starting with a DNA or an RNA at a pure quantity that does not exist in nature. I want you to imagine the worst case scenario for how bad this is, because it is that bad. They do not find a single contiguous sequence in the wild, not a single one. What they find are fragments, and then they assume that these fragments represent what they call the whole. It would be very similar to finding a small scrap of a book outside, and if it fits into the Encyclopedia Britannica somewhere, then you say that the Encyclopedia Britannica was in your backyard. And if you choose the right kind of book, or you choose the right kind of abundant background signal, then making that claim could just be a wholly manufactured lie. And so one of the possibilities that happened in the pandemic is that there is a loud, incredibly loud background signal up there, and they have represented that as a very high fidelity signal that they can differentiate from a background when they really cannot. And in fact, most of coronavirus biology is based on this concept that they find a broken, fragmented signal, they assume the whole signal, which is again based on fragments that were found earlier, I guess. I mean, if you try to trace this all the way back, there's just really assumption after assumption after assumption. And if in 5 years they have added 5 more genes to the coronavirus genome because they discovered them now, it'll be very dubious, just like this has been so far to assemble. And you can go all the way back. I mean, I've tried my best. Some of the books that I've gone back to are books that are just thrown out of libraries that are coronavirus meetings from the Netherlands and other places. These phenomenon are real, but how they are represented, and more importantly, how they have been represented in the past, say, decade or so, is not. And so that's very important to understand, because as an academic biologist, it has been tricky for me to juggle the idea that that all these people can be so wrong. There has to be a way to reconcile most of the observations that we've made in the past and most of the observations that are being made now with what we really understand in biology. It can't just be that everything's a mythology and, all viruses are fake and all disease is fake and all health is fake and all, and there is a tendency, I think, for the operation that we are in to tempt us with those kinds of oversimplified. I like to say you're tempted with two different things. You either have to accept a stupid, complicated explanation that you're too unsophisticated to critique, or you have to accept a dumb, simple explanation that is just, obviously not right.